Globalization and international travel have contributed to the worldwide spread of infectious disease. Scientists at Los Alamos National Laboratory are addressing this serious national and global threat with a unique technology that would detect infectious diseases, specifically bacterial ones, quickly, simply, and efficiently. The Universal Bacterial Sensor.
To diagnose infectious diseases quickly, we must be able to identify the disease-causing organisms or pathogens. Nature has provided us with an excellent example to show us the way: the human immune system. Our innate immune system is capable of recognizing all pathogens. We have tried to mimic this recognition in the laboratory to develop a universal strategy for the diagnosis of all infection. To develop such a universal bacterial sensor, we must first begin to understand this recognition and the interaction between the pathogen and the human body. Bacteria are covered by a cell wall or a cell membrane, which are comprised of, among other things, of greasy lipidic molecules. Some of these greasy lipidic molecules are secreted in our body during the course of an infection. These are the molecules that our human innate immune system recognizes very early in the course of an infection, even before symptoms are observed. Detecting these greasy, oily biomarkers and watery aqueous blood, like oil and water, they do not mix. This makes it a challenge to detect them in the laboratory using conventional methods that were originally made for detecting water-loving proteins. Achieving our goal of universal bacterial detection required an understanding of what nature, that is, our human immune system, does to circumvent this problem.
The human body transports molecular signatures, also called biomarkers, using host carrier molecules. These are called lipoproteins, and they're actually made up of lipids and proteins, like a biological taxi, taking bacterial biomarkers from the site of an infection to the immune system. We developed two novel methods for detecting bacterial biomarkers directly in blood. These methods comprise the Universal Bacterial Sensor, which is the first approach to diagnose all bacterial infections in a drop of blood. To actually measure the biomarker trapped inside these carriers, we have to release it. We developed a quick and easy sample processing method that takes less than 2 minutes and can be performed easily by a non-expert.
As part of our platform, we use antibodies attached to fluorescent molecules to show the presence of the biomarkers that we're after. The fluorescent portions are quantum dots. Many different quantum dots can fluoresce when excited with a single laser wavelength. This allows us to measure several distinct biomarkers in a single sample. These properties have led to a straightforward multiplex detection assay. This method works for all types of bacteria, gram-positive, gram-negative, or even gram-indeterminate. With no prior knowledge of the bacteria, our system can discriminate the nature of the pathogen within about 30 minutes.
We have applied this technology to provide fast and reliable answers in many research applications. We have worked in concert with medical and veterinary clinicians in order to evaluate animals in research studies and during vaccine and clinical trials. In fact, we've tested our tuberculosis assays so far on humans, cows, and badgers. This sensor and the information that it provides changes the way that we think about immunology and infectious disease research. The Universal Bacterial Sensor is the first method that measures these greasy, oily biomarkers without compromising their biochemical properties. Our work also indicates the importance of the role of these greasy, oily biomarkers in infectious disease and the role of host carrier lipoproteins in this association. These methods have far-reaching applications beyond infectious diseases to the study of other conditions such as cancer.
The Universal Bacterial Sensor can identify pathogens easily, quickly, and at the point of need. The sensor retains its sensitivity to identify the pathogens even as the bacteria evolve, such as acquiring antibiotic resistance. With our long history of providing innovative solutions to national security challenges, Los Alamos National Laboratory has developed the Universal Bacterial Sensor to identify and respond to infectious diseases, whether in a remote village, in a clinic in the United States, or with our troops deployed around the world.